Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome to the Genetics Podcast, everyone. I'm really excited to be here today with Dr. Marco Quarta, who is the co-founder of multiple companies in the cellular aging space, in particular, one most recently called Therapeutics. Prior to that, he was on the faculty at Stanford, where he was a postdoc previously and did a lot of work in the field of aging and cellular senescence in particular. We're going to talk today about the way that our understanding of aging has evolved over the past decade, and also Marco's transition from academic research, where he used to spend 100% of his time, into building companies where he spends 99.9% of his time now, but still does find time somehow to teach students and stay involved with the university. So Marco, it's a real pleasure to be here, and thank you for joining me.
Thank you, Patrick. The pleasure is mine. I'm really excited to share some of my story with you today.
You founded your first company in 2018, but prior to that, as I mentioned, you had a long career in academic research. Could you talk a little bit about what you were focused on, and then what drove you to actually found your first company and then subsequent ones after that?
Well, yeah, actually, technically, my first company was way back. It was in 24 in Italy. Wow. Yeah, where I'm from. I was still a PhD student when I started my first startup, where I was the CEO for a couple of years, and then we did a joint venture, and the company still exists and up and running in Italy. And that was my first experience. And I did another one in UK afterward, prior to come to US. So I've been involved in several organizations, all in the field of aging or regenerative medicine. And that's kind of was been my interest for a long time. Actually, a very young age, I decided I wanted to understand and change the aging process. Actually, this is an early inspiration of mine as a young kid, which I never abandoned. Clearly, I'm still on this. So all my research, academic research, and more applied science has always been aligned on that mission. And that's why I'm still doing it.
How's our understanding of aging changed in the past decade or two while you've been in the field? What do we know now that we didn't know a few years ago? And then what are the big open questions still as well?
Yeah, I mean, definitely aging is a very complex biology, right? It's a multifaceted phenomenon. So the more we dig down and understand, the more we think we realize that we start to scratch the surface. But I think we also understand how little we know. And something that probably, I think, changed in the past 10 years is shifting from the model of hallmarks of aging, which was kind of widely accepted, which is a reduction or a very simplified view of how aging is, which kind of worked as a model, but was also borrowed from the hallmarks of cancer. That's really that was this translation. I was never really happy with that model. I think it's still used, but the truth is, as we keep refining our understanding, we realize how, number one, the hallmarks are all interconnected and one leads to another, some are upstream to another. For example, like if you look at how DNA damage versus telomere friction or epigenetic instability or mitochondrial damage that are at one level, but they all are integrated downstream to induction of cellular senescence, that may be downstream to that, and how that one leads to the other. And then there's more and more, let's say, biology that we're starting to learn that makes this model more fuzzy. So I think we're moving away from this, and I'm happy about this. I mean, it's still easy to explain things, but probably not a good working model. I better prefer a more integrative view of cell aging, where things are really like modulated on a large scale, and there are shifts in this network in the regulatory process. They understand maybe where certain nodes gets a higher weight versus others. That might also leads to a different trajectory of how a cells can age and then a tissue. As a matter of fact, we all age at different rates. Even within the same person, each tissue or organ can age at different rates or in different ways, driven by different aspects of this biology. So it's complex, but I think we start to know enough to take advantage those as potential therapeutic opportunities.
One of the things that you mentioned previously is how we often probably have this misconception that maybe every cell is aging at the same rate. And you kind of alluded to this before. But I think that's not the case based on what you've said. How do we get a clearer picture of what's actually happening within a tissue or in a cell? Does single cell genomics and other kinds of these relatively new technologies help with that? Or what do we need to do to understand that picture a little bit more?
No, yeah, that's a great point. For sure, we have an technological advancement in genomic technologies, which is key. Maybe just focusing on one of the things that I'm working on as you mentioned, cellular senescence, which is the focus on Rubedo Life Sciences. And I have to go back to what we were saying, how cells can age and take in different trajectories is one thing, and that can result in different, let's say, pathological manifestations. One is undergoing into the cellular senescence, say, programming or phenotype. But even that, like if you look at senescent cells, this is highly heterogeneous cell type. There is not such a thing as one senescent cells, like there is no such a thing as one cancer cell. There's a highly heterogeneous. Actually, the two are a little bit connected. One is the other side of the coin somehow. Senescent cells is a tumor suppression mechanism. And cancer cells, many share still features of senescence. But beside that, that's one type. It really depends on how are the damage, the stressor, the stimuli that induces senescent cells to become as such can lead to different flavors, if you want, or different aspects of different regulatory or compensatory pathways that leads to senescent or senescent-like states. So they're not all equal. They're not all respond to the same targets. For example, in the context of a target discovery or drug discovery, it's really important that we know what to go after, because you might develop a beautiful senolytic that is useless in the wrong tissue, in the wrong indication, clinical indication. So how to address this? Well, certainly single-cell multi-omic technologies has been an incredibly valuable tool. And to address that, for example, at Rubedo, we built a discovery platform based on that. And it's called it Alembic. Is a platform that works at integrating multi-layers of single-cell multi-omics technology from single-cell RNA sequencing or ATAC sequencing or spatial multi-omics, just to name a few. And we leverage different forms of math, which artificial intelligence like machine learning or deep neural network, trained, for example, with a large array of proprietary libraries of models of senescence. And so then you can take clinical samples and ask questions like, are there any senescent or senescent-like cells in this disease state? And if so, which ones? And then what's special about them? What are the vulnerabilities? How can we describe them? Can these be biomarkers? And what should we target, etc.? Because there are so many differences. And I give an example. So even just to take a cell in vitro model and you induce senescence with the same modality, let's say chemotherapy-induced senescence, it's widely accepted that the resulting state is senescent cells, which you do. You look at typical markers like beta-gal or EDO incorporation, they look all equal. But when, for example, we treated the same cell type induced with two different types of chemotherapy with standard of care, resulting something they look the same senescent cells, but then treated with the same compound, which is a BCL2-XL inhibitor, navitoclax, widely described senolytic agent in certain cases. This is exactly the case. So in one case, this clax showed to be a very good senolytic, but in the other case, very poor. And when we did analysis, we look at the transcriptome landscape overlap or pathways analysis, there was very, very little overlap to none. So Same cell inducing senescence with the same modality, chemotherapy, but two different agents. You get two different types of senescent cells, one that responds to this target and one that it doesn't. And more important, when we start to look at patient-derived primary senescent cells, for example, we saw at high variety in certain diseases, this target is completely irrelevant. In others, it is. So that's why I say it's so important to leverage those technologies so you can really support and inform a drug discovery that is more meaningful.
Yeah, interesting. And so it sounds like, playing that back, that across any disease, having a better understanding of if cellular senescence is at the core of the issue, or at least part of the issue, it's not enough to just know that cellular senescence is part of the problem. You really need to understand. If we take liver disease, for example, there may be different ways, that different insults to the liver or different mechanisms by which you can have cellular senescence. And then the way you treat those as a result would be different, because you've arrived at that state differently. And so there's something, there's some phenotype beneath the top level one of senescence that is actually more important, and it impacts the way that you might treat it successfully. Have I got that right?
Yeah, no, that's absolutely right. And even not just different states, but also different transitional states. So I think the pre-senescence, the deep senescence, primary senescence, secondary senescence, one can be rescued. The other is too late and it's really need to get rid of. One can be a consequence or a catalyzer of disease progression, but others could be causal. And these are all very important questions that we need to address. As I said, cellular senescence is an emerging field. I think there's so much more to do. There is a need of way more modalities and therapeutics that can be developed. And I think in the years we will see, and we should see, hopefully, see a growing arsenal of therapeutics to really address diseases in different ways. In the future, more than a reactive medicine, maybe even in a preventive medicine fashion, maybe in a combination therapies, as we learn more and more about this biology of how different therapies can be applied together. So we're really at the very early days.
Yeah, and it's one of the things that is both exciting but also challenging, I think, about taking the approach that you all are, about going at some kind of core part of biology here. Is when I look at the Rubedo Life Sciences pipeline, you're going after dermatological diseases, pulmonary, liver, neuro, musculoskeletal, oncology, because aging is at the core of almost every major disease category. There's obvious benefits to this, because you're understanding a part of biology that helps you to treat so many diseases. But how have you found that to be challenging in the company building side of things, and needing to be fluent across all these different organ systems and areas of biology?
It's certainly challenging, especially in early stage companies. You can't do everything, even in a late stage global biopharma. So you need to be very strategic, and we are. So we built that pipeline. We did exploratory work, which is easier to do, but of course decided what to advance and prioritize is key. And so the lead program is in chronic inflammatory skin conditions, because we found that as a lower hanging fruit. So something where we can move faster. It's easier. It's not easy at all, but it may be easier compared to an idiopathic pulmonary fibrosis population and disease. From a clinical trial perspective, you can compress timeline. It's an organ that you can see and watch and sample, and just allows you to do things in a different way compared to other organs. Large number of diseases that have common denominator could be the cellular senescence, even though they're very different diseases. Chronic psoriasis versus chronic atopic dermatitis, they are initiated in a completely different ways. But senescent cells can really be sort of the glue that drive the persistence of those lesions and this chronic inflammatory process. So great place to start to validate the target. This is a first in class. There's novel targets that we are validating in this biology and therapeutic. First validation of our approach. And so we thought that's where we can probably go first, and it allows even to spin into other parallel directions like cosmeceutical or skincare. And that's why we announced recently this partnership with Biostorf, the company behind products like Nivea, Coppertone, Eucerin, etc., to really treat not a diseased skin, but a healthy skin in an aging context, which is another way to ask if and how this biology plays a role. And that's just an entry point. But from there, then we can develop, and we are advancing the other programs, which are different targets, different molecules. So completely separate modalities for lung diseases, and working with strategic partner, and to build other verticals on other diseases, like you said, liver or etc. But it's a journey. So this is like we build a roadmap to start and grow from there over time. Of course, we're not doing everything at the same time in the same way. It would be daunting.
And how there's another vector of complexity that I guess you've had to make some decisions on, which is the therapeutic modality. You can do small molecules, you could do ASOs, you could do gene therapies, you could do gene editing, you could do biologics. And when you have a kind of platform biology approach like this, it can be, again, kind of challenging because you have a wide swath of tools that you could take to this, right? Have you made some decisions around we're going to focus on small molecules for now, or have you actually gone the opposite and said we're going to partner with people who understand these therapeutic modalities and bring our expertise in biology? How did you think about that?
Yeah, no, this is exactly right. So we decided to focus on small molecules as a first modality, which this is what we are moving forward. Our RLS 1496, which is the therapeutics that they're going into the clinic soon, is a small molecule. But to your point, there are other modalities that we have been looking at exploring, not necessarily to do all in-house, but with partners that have that expertise, so we can join forces. Is a win-win. We provide that expertise and unique understanding on how to find and target senescent cells, while partners might bring a certain drug discovery pipeline, so that can be built together, because otherwise, we will have to build a whole new team inside dedicated to, let's say, biologics to work in parallel with one molecule. Possible, but not at this stage.
Yeah. I wanted to just read a couple sentences from your website, because I think someone has done a really great job. I don't know if it's you or someone in your team, just writing this and kind of clarifying it down to the core. And then I'll just ask a follow-up question off it. But the website says, as we age, our cells become less functional because they accumulate errors. These errors, such as epigenetic changes, result in cell damage over time. Eventually, the cells become pathologic and cause diseases. I'm paraphrasing and cutting out a few points here. Among these pathologic cells are senescent cells that drive age-related diseases. Senescence is the process by which a cell ages and permanently stops dividing but does not die. Any cell can potentially become senescent. Over time, large number of senescent cells build up in tissues throughout the body and release toxins. And when they reach a critical mass, they cause chronic inflammation, fibrosis, tissue degeneration, and drive many age-related diseases. I think this is a really elegant summarization of the process. And it makes me realize that there's many steps in this process that you can intervene. At the very beginning, we say they become less functional because they accumulate errors. What if we stop those errors in the first place? Then there's the stage at which you start to accumulate senescent cells in an organ. You could probably detect that and maybe arrest that process or reverse it. Anyone listening to this can imagine that there's a lot of different ways, upstream or downstream, to try to right the ship. And I'm curious what you think, what you all are approaching this. Is it at one particular level of that causal chain, or are there hypotheses that you may need to intervene at different levels of the chain in different diseases? I'm just curious to hear about how you break down the problem from that really elegant kind of top to bottom cascade that's described.
Mm-hmm. Yeah, it's more the second option. I don't think there is one way to look at this. And that's why, again, going back to understanding the biology, how that plays a role, you might need to have interventions at different stages of this cascade. Also, because it depends on what cells and what type and how they're really playing or interplaying with the specific dynamics in specific diseases. Give an example, like as I said, like in the skin versus let's say in the lung, and just keep it high level. But like in skin diseases, in general, again, there are exceptions, but in general, let's say in this chronic skin inflammatory condition, something caused the lesion, the irritation, the damage, and the insult that results in a skin lesion. It could be, if it's a rosacea versus atopic dermatitis, these are different reasons. It could be an agent, an irritant, it could be genetic, autoimmune. But the fact that there is now those lesions persist and become chronic, there might be that's a formation of cellular senescence during this process that for some reason is causing a maladaptive response to the immune system into the tissue, prevents the lesion to heal, prevents immune cells to do what they're supposed to do. And they contribute to this persistence, not necessarily the disease formation, but it's really the disease progression, so chronicization. So you need to know how they're playing and what to target on this cascade, and maybe get rid of them. So you release immune cells to go back to what they're supposed to do, which is healing instead of like breaking the tissue down. That's an example. Maybe it can be very different in, let's say, in IPF, idiopathic pulmonary fibrosis, or interstitial lung diseases in general, where we are at the very top of this cascade of events. For example, IPF, there is a genetic predisposition, generally a familial mutation that can act on some forms of genomic instabilities, like telomere friction. It could be a mutation on shelterlin and TERT, etc. But the result is, so that's your baseline. If there is an insult, it could be like cigarette smoking, virus, and pollution, whatever it is, drives cells into premature and stable senescent state because of this genetic instability. Healthy people can recover from that. So there is this cycle. But in IPF, the first cells that undergo this are progenitors of the alveolar tissue. So, ATQ cells and other cells or basal cells that undergo this senescent state. And then you get these two problems. On one hand, you deplete the self-renewal capacity. So cells that actually are responsible to repair the tissue following this damage. On the other hand, you accumulate cells that instead persist and acquire this senescent state and start to secrete factors that are shifting and informing the immune cells, the immune surveillance coming to the lung to change behavior. And together, they exacerbate this process. And eventually, at the end of this cascade, they recruit myofibroblasts. They transition into, fibroblasts. They transition into myofibroblasts. They start to secrete collagen and more TGF-beta, where that becomes really a feedback loop. Eventually, we have scarring and so fibrosis. And it's a slow progressing, growing, degenerative process. In this case, it's very different cell types, very different biology, but it's also at the very top of this cascade. And so you need to do an intervention that is very different to have a disease-modifying therapies. In this case, you can probably stop the progression. So importantly, you're not really treating fibrosis. You're treating what's causing the disease to progress. And by doing that, you can probably enable the remaining healthy progenitor cells to go back and repair, to a certain extent, immune cells to support them in this process, and overall slow down this cascade and this progression. So really improving quality of life and giving more quality of years in a disease that is really terrible, where standard of care today has very minimal efficacy. Has some, but only some patients, not all, respond the same way, but definitely doesn't improve the quality of life. And it can slow down the fibrotic formation a little bit, but certainly not curative. So high unmet medical need still.
Yeah, fascinating. One of the things I wanted to talk about as well is another company that you founded, co-founded, or actually involved with, which is Turnbio, that is in the same space, but it strikes me as almost approaching the problem from an orthogonal direction. So a lot of what we've been talking about here is really detailed understanding of the mechanism and how different diseases and different organ systems cascade over time. Whereas the approach with Turnbio is to say, okay, we learned from induced pluripotent stem cells that there are actually some transcription factors that are incredibly magic in a sense. We can do things with them and they have dramatic impacts on cells. And I think you've taken a similar approach to look at mRNA delivery of a really special set of transcription factors that essentially can kind of turn back the clock on aging, so to speak. And I'm under no illusion that this doesn't also require a deep understanding of the mechanisms underlying disease, but it's it seems to me like a really different approach. You're saying, can we do something that's unnatural in some way to turn back the clock versus understanding the natural process and then intervene? So maybe you could talk a little bit about the origins of Turnbio and what you're working on there too.
Yeah, no, and I think you nicely summarize. Really, this is also my vision. Yeah, this is a work that also was one of my the focus of my lab at Stanford, work that I was doing with my friend and colleague Vittorio Sebastiano at Stanford. And we were really exploring the question as well, if we see this in nature, right? Like during fecundation, during reproductive process, in reproductive biology, where you have two old cells, differentiated cells, germline, oocytes and the sperm, from older individuals that carry, some signature of aging, but the outcome is a completely age zero offspring. Not trivial point, because you say, well, sure, that's how it works, right? But that's not obvious. If we think the first clone, like Dolly, right, it was a nuclear transfer, took a somatic cells, take the nucleus from skin cells, fibroblasts from the mother into the embryo and transfer that and then implanted that. Dolly was born, great. But what people know less, that also this aging signature was transferred. And Dolly somewhat presented some prematurely forms of aging. So there is something that happens in natural reproduction that can erase that and reset that. And we found the same happening during induced pluripotent stem cell process. So somehow recapitulate that. So we're really asking the questions, can we understand and sort of use this to kind of activate this machinery, whatever it is, that can reset this epigenetic clock related to functional biological aging without de-differentiation, so without pushing cells back to change identity. So if you take a skin cells or a brain cells will stay the same. You don't create an embryonic cells that certainly you don't want in your body. It makes teratomas, right? And so we found that doing this transient, especially using RNA, which is not integrative, you can pulse it, and we developed a technology to do that and also to be targeted. That's a different platform that is at the core of Turn. How to target specific cell types in different tissues to deliver those factors. But the answer is, you can reset this aging signature. We look at this in many ways, epigenetically, transcriptomically, functionally, and the cells reacquire functions like a younger version of themselves. We did this in human cells from human donor. Very interesting experiment where we transplanted in immunocompromised mice stem cells from, in this case, muscle stem cells from human donors from the age of 10 to the age of 85, 90. And we applied this prior to transplant, this epigenetic reprogramming approach, ex vivo, so taking the cells from a donor, rejuvenating them, quote unquote, and then transplanting them and measure their regenerative capacity, engraftments, functionally the muscle, if it was like doing physiology, like force production. Is it getting better? Is it less fibrotic, stronger, all those things? And what we found is at any age, there is some improvement. Even if you take a 10-year-old person and you apply this, it gets a little bit better in doing this, but not that much. But, something is still happening. The major delta was after 65. That's really where we saw a dramatic change in poor function versus gain of function, which interestingly, 65 is the average. There are lots of exceptions, but it's average age where we start to see this exponential risk to develop year after year age-related diseases, right? Not one, but all of them. So there's something there that is biologically age-specific. So that's kind of the idea, right? Can we do this in vivo or just ex vivo? Can we restore function where cells are losing it over time? And importantly, somewhat prevent those cells to become pathologic. So to put it simple, if Rubedo is focusing in removing the bad cells, the bad apples, before it makes the other apple being rotten as well. So you do this and keep the apple healthy. The good apples, if you don't do anything, we will still be, going to this path. At some point, you find more rotten apples. So Turn wants to do the other side, want to prevent the good apples to become bad. And so that my aspirational vision in the future, that those two therapies could really work synergistic in the future towards a really more longevity medicine concept, where now they were both focused on therapeutics as a first step.
Yeah, that is fascinating. Are there particular diseases where one of those sides of the coin is more important than the other, where, policing the bad apples and removing them is most important. And then the rest takes care of itself, or the reverse, where actually, yeah, I don't know for what reason, you're the expert on this, but do most diseases really need both to cure them dramatically, or are there somewhere you may only need one and then the body takes over the rest?
Yeah, I think therapeutically, there are certainly diseases where one is an intervention needed and maybe the other is less, although one can support the other. But I would say, to go back to my previous example, in idiopathic pulmonary fibrosis, senescent cells is a well-recognized, established phenotype that is really part of the problem. So somewhat, if you can find a way to correct the genetic familial mutation that causes genetic instability, it's a way to sort of go at the core of the problem. But during the disease, once you have it, removing senescent cells is probably the best intervention to kind of stop or slow down this cascade. And there's an example. That's where those senescent cells are like building up is somewhat inevitable. And the more you get them, and it maybe goes accelerated. So it's very conducive of creating this like harsh, toxic, pro-inflammatory environment. And so you need to clean this up. Otherwise, no matter what you do, the immune cells and stem cells are not going to be able to work properly in this pro-inflammatory milieu. On the other end, maybe there are diseases that are more driven by lack of regenerative capacity.
Right.
And that can be from different form of, let's say, cellular aging. Simply stem cells are kind of, losing their stem cell self-renewal capacity. They're depleting the pool. They're taking a different fate, differentiating into something that maybe is more profibrotic versus their lineage. So that's where you might want to kind of reprogram the cells and boost their regenerative capacity. So wound healing, just to make an example, could be another situation where you want to kind of prevent the tissue to degenerate and rather inform and reprogram the tissue to go back and work in how it used to do when it was younger, biologically. And say biologically because there is, when we talk about age-related diseases, it's not necessarily a problem of the elderly. It's certainly a problem of the elderly. But you might be biologically younger and for whatever reasons have a prematurely biologically aged tissue that drives those diseases even decades sooner.
Yeah.
And we see this in cancer survivors, like standard of care is a dramatic way to drive cellular senescence, for example, chemotherapy, radiotherapy, immunotherapy. I mean, even pediatric cancer survivors, children with leukemia, they develop age-related diseases in their 30s, 30 years sooner than the average person. More cancer, cardiovascular diseases, of course, skin, everything looks aged and is measurable in this way. And the markers of senescence inflammation. COVID-19, SARS-CoV-2 is another example. We see in the clinics people in their 40s coming with the need of lung transplantation instead of in the 70s because they have something that looks like a 17-year-old interstitial lung disease like phenotype. So it's biologically speaking, when the tissue is aged, but they can, again, we were saying earlier, take different directions. In one case, maybe reprogramming and boosting the regenerative capacity might be the key to prevent degeneration. In the other case, removing a source of pathologic factors like senescent cells can, enable the immune cells and the rest, the regenerative capacity to shift back into a more active function and be a therapeutic opportunity.
Yeah. I was curious to get your take on whether there are kind of core therapeutics of aging that have potential. Like, we know that people take rapamycin, for example. I think there was a Lancet article relatively recently that said 95% of people who take it off label are doing so for healthy longevity, anti-aging reasons. And I think there's there's something beautiful and powerful about finding something that reduces all cause aging or can have a system-wide effect. I'm just curious to get your take on where the science is with that kind of core aging pathology, if we can call it that, and what you think about that as a general paradigm versus needing to really understand on an organ tissue by tissue level versus searching for something that could be system-wide.
Well, yes, let's say my ethic is that I follow prevents me to comment on a broad, wide use of potential geroprotector drugs or medicines without a deep understanding and test through a clinical trial process. The reason is, while the perception that some of the interventions, let's say rapamycin or metformin, others, and perception created from preclinical studies and some of this anecdotal clinical outcome, etc., which I think is insufficient to tell us how, when, and if you should take those. Some people may benefit, no doubt. Other people might actually have completely no effect. For some people, it might actually be detrimental. And because what you were saying earlier, it depends on your biology and where your aging bias are somehow, that you may need to take different interventions or even use the same, say, drug in different ways with different regime treatments, etc. So this biohacking idea could backfire, especially in the long run. And I would always be hesitant, even with supplements. You just play with something that for you don't know how that responds. It's even metformin, right? That it looks like so very safe drug. It's been out there for decades, and we know very well how it works. But most of the data are from diabetic people. That's not the same biology. The other people that are on a different side of the spectrum. And some people will be closer to a diabetic-like metabolism. Others will be completely different. Obvious example, and there are studies showing it. You change your metabolism now by pushing with certain very intense active exercise that counter certain pathways, and where taking metformin can be, for example, counterproductive. And it can actually be counterproductive also against the benefits of this exercise. You're just dealing with two different metabolisms. So we have to be careful. And that's why we need, I know I wish we would do enough to jump into conclusions and say, let's start taking this or that. I think there is lots of perception of what things can, work, how we think it is, but these are speculations at the moment, and they're not supported by enough science. And, that's a very important point to me. And this is one of the topic at the Friedman Institute, this nonprofit scientific society that I founded with a number of key opinion leaders in the aging field from both academy industry. Some of the most relevant names out there. And one of the goals is really raise a scientific bar and make sure that we focus on high and deep science and prevent like pseudoscience or anecdotal science, if not even snake oil, to interfere with the scientific progress of these therapies. Fortunately, it takes time. There's a lot of things we still don't know. And I think we will address those questions, but it takes time. So biohacking can be risky.
Yeah. And I guess the risk is somebody gets hurt, something happens, and it could set the field back quite a bit, right? Besides there's the risk to the individual, but also the risk to the field as a whole, right?
Absolutely. It's just one accident and we will, and 10 steps back.
Yeah. You'll have to talk about it for years. Yeah.
Yeah. We know nobody's going to benefit from that. So I think we just need to be patient. We're doing amazing progress. The field is growing. There's an ecosystem really supporting this that we never had before, but it takes time. And we have to take into account that there's a trial and error. There will be lots of failures in clinical trials in progress, but this is how it is. How many failures we had in oncology before the first therapies, and after 50 years, we're still working on it. And there are still lots of failures, but lots of progress, and the field changed medicine. And now we can say we can treat and cure cancer. It's not a done deal. And aging is even more complex than that in certain ways.
Yeah. As we wrap up here, you have a track record of being ahead of the curve multiple times. And so I want to ask you one of my favorite questions, which is something that you're really excited about now. It could be, it could be a tool, it could be a new area of science, it could be an idea, something that you think isn't quite mainstream yet and people aren't talking about it, but you think is going to only become more important in the next couple of years. Is there anything that springs to your mind that you would just want to shine more light on right now?
Well, maybe something that maybe the opposite, something that people are talking too much about is AI.
Yeah, perfect. Why?
I think there is an overhype of what AI can do or will do and will solve, especially in this field. I don't think that's okay. I think it's an amazing tool. I think it's very helpful. It's not going to be a magical bullet. It's not, at least not anytime soon. But it can be extremely helpful. It also can be extremely confusing, introduce bias that can lead the field in a completely different or wrong direction. So we need to be very careful about it. But it's also a very important tool because it enables to integrate so many different aspects and technologies and readouts that we now have as technology are developing that otherwise would be impossible. But we need to be very careful how to build them, how to train them, how to use them. And because it's not like you press a button and magically you get answers. It's just another tool to connect the dots. And I think it's very effective, but also a little bit over empowered at the moment.
Yeah. I think most people would agree with you on that. There's so many expectations about what it's going to do that it's all priced in, right? If it doesn't quite deliver on absolutely cracking every problem in modern biology, some people are going to be disappointed.
Yeah.
Well, Marco, thank you. This was really great. I learned a ton. You've been very busy the past couple of decades doing some great science and building great companies. So thank you for sharing all these insights with us.
Well, thank you, Patrick. It was a fantastic conversation. I really, really enjoyed your insights. And I hope, my 2 cents can be of any inspiration or help with others. And definitely this is a field that needs more and more contribution, more minds, intellect, talents. And we're really at the beginning. So, the more, the better.
Yeah, couldn't agree more. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selective Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.